Good. And next, we're moving on to Rebecca Charnock, who's joining us from over in West Wales, somewhere near Aberystwyth, I think. So Rebecca is the Industrial Research Development Manager at AberInnovation, which is the Innovation and Enterprise Unit, part of Aberystwyth University, involved a lot in agri-tech and general technologies, but particularly today, she's going to talk about circular economies and sustainable systems and some of the things that are being researched over there. So thank you very much, Rebecca.
Thanks very much. Thank you, Adrian, and good morning, everybody. I'm just going to share my screen and bring up the presentation. This morning, I am going to talk to you both about our new campus and also about the excellent facilities we have here and the research that we have inside that campus as well. So just move on to the next slide. So I guess I should start by mentioning a bit about the campus. We are a tech owl company, and we are, let's just sort my screen out, I can't quite see everything here. Right, so we're a subsidiary of Aberystwyth University, and we have a fantastic brand new building just completed this August, providing new facilities and equipment, and it is our aim to use these facilities to support collaborative companies to deliver innovative products and services, and all with a feed, food and fuel theme, working with expertise from Aberystwyth University. So this capability has come to fruition through investment of £40.5 million. It's been mainly due to European funding through the Welsh European Funding Office and funding from BBSRC. Now, this has helped to provide us with a fully equipped facility co-located with one of Aberystwyth University's two campuses, and where we can provide access to academic expertise, and all with the aim of accelerating collaborative innovation. So, and as we are also known, Aberystwyth Innovation Enterprise Campus, or AberInnovation, we form one of 9 UKRI funded innovation campuses, and it's nice to see too that we are filling a gap in this region here. So our main aim is to enable companies to invest, test and validate, and to provide a space to innovate in a low-risk development environment with academic expertise available to advise and support. So AberInnovation is made up of Biorefining Centre, a Future Food Centre, and an Advanced Analysis Centre, as well as an Innovation Hub. So to go into a little more detail on each of those centres, the Biorefining Centre consists of a primary processing unit, downstream processing unit, which can be food grade, depending on what the need is, and a fermentation unit, which can also be food grade. Just upstairs from these is the Clean Lab, which is ISO 7 standard, so suitable for pharmaceutical and cosmetic grade activities, and a low carbon laboratory, which will contain a number of growth chambers. So these then link to a bioprospecting lab and synthetic biology lab based in the Advanced Analysis Centre. And this is just an overview here, plan. So Biorefining Centre at the back here. In red here we have the Future Food Centre, and obviously the new unique selling point for our centre is that these food grade labs can connect with each other. So what does the Future Food Centre consist of? So we have a dairy and liquid processing unit here, and a meat and solids processing unit. And these are all food grade labs. So we also have packaging and storaging facilities, food testing laboratories, including microbiology labs, within our Advanced Analysis Centre upstairs, so they can connect with the Future Foods Centre, and we have temperature control area and demonstration kitchen as illustrated here, and a sensory testing suite here with sensory testing booths, and we will have a sensory testing panel as well to use those. So the Advanced Analysis Centre, which is upstairs in the building, contains a synthetic biology lab, as mentioned, a bioprocessing lab, which Biorefining Centre can also integrate with, and as mentioned before, the microbiology lab as well, which the Future Food Centre can integrate with. We have autoclaves, media preparation lab, we have principal laboratory areas where there are high performance liquid chromatography, gas chromatography, and mass spectrometer areas, as well as access to data processing. So all of these centres provide a plethora of equipment and facilities for industry to come and use and to collaborate with expertise. And of course, at the front of the building, really important to help with those collaborations, we have an innovation hub. And in that hub, we have boardrooms and seminar spaces, meeting rooms, an open plan office space as well for desks for industry, and for academic partners to come and meet with them as well, and to hot desk. And the idea being that there should be a hub of activity with academics meeting up with industry within these spaces. So we also have across the road an office space which has 22 individual offices, and many of these are occupied at the moment. So we have membership packages ranging from full resident and associate membership, which allows companies to use the office space on a part-time basis, and a virtual membership. And we have companies take up a membership in order to be close to academic activity, and it can also allow them to tap into Welsh Government funding to assist with research and development where they require support from academic expertise. And this is just 21 members so far, and their logos, some of their logos of the current members. So quite a range of companies, mostly fitting in the feed, food and fuel theme, all with industries that support these themes too. So just very briefly, just a quick slide just to show, just to illustrate some of the areas that we have expertise and capabilities in. I'm not going to read it, just leave it there for you to see. So ranging from looking at farming and food production, towards product quality, and all the way to bioactive discovery and extraction processes. So a plethora of expertise and facilities there. So I'd like to just mention two programmes that are running within Aberystwyth University, which link closely with our AberInnovation facilities. And these programmes provide support for industry to tap into, and this includes access to expertise within Aberystwyth University and using the facilities in AberInnovation. These programmes support small-scale collaborative projects that focus on new innovations, products, services, and improving processes. So often these projects can lead to or can act as a feasibility study towards bigger collaborative projects between industry and academia. And I should point out as well that both programmes are European funded and carried out in collaboration with a number of partners. So one of these programmes is, as illustrated here, the Beacon programme. This is a partnership between Aberystwyth University, Bangor University, Swansea University and the University of South Wales. And the Beacon programme at Aberystwyth involves researchers and academics based in our Institute of Biological, Environmental and Rural Sciences. They've developed the programme by building on their history of R&D projects utilising a range of feedstocks and conversion systems. So the Beacon team's laboratories provide a flexible pilot scale facility, and these are now based within AberInnovation's brand new Biorefining Centre. And the Beacon team's main aims are to work with industry to support the development of innovative, economically viable industrial biotechnology processes that will create value added from products from waste. So we also have a Future Foods programme, which is a collaboration between Aberystwyth University, Economies, Food and Drink Wales, and aims to help the Welsh food industry tackle global challenges and understand global best practice. And it does this by engaging with industry in collaborative R&D, and this R&D helping industry to understand and address changing consumer demands and to provide support for them to produce new and innovative projects with sound scientific basis. And the programme's research capabilities span the whole of the food value chain, from plant and animal breeding and agriculture production systems to human studies, exploring health benefits of novel foods. And along with AberInnovation, Aberystwyth University research farms and Aberystwyth University expertise in areas such as food, raw material bioprocessing, advanced composition analysis, and clinical trial facilities. This provides a breadth of capability unique within Wales. So I thought I'd tell you a bit about some of the projects that these programmes have been involved in, and to illustrate some of the areas which industry can work with AberInnovation and link up with our Aberystwyth University expertise. So I'm going to discuss a bit or let you know a bit about the research on agricultural waste, and in this case wheat straw to produce a sugar alternative called xylitol, and also a research project looking at food waste, finding value in food waste, and in this case producing taste enhancers from what was food waste. And finally, finding a more cost-effective source of pharmaceuticals, and in doing so, adding value to farmland as well. So, agricultural waste. So this example is looking at producing xylitol from wheat straw. So why wheat straw and why xylitol? So there are approximately 14 plus tonnes of wheat straw per year produced in the UK, and of this, 30% is used for animal bedding and 40% sold off farm, again used for animal bedding. The remainder, so 30% remainder, is chopped up and returned to the soil. Sorry, it's 40%. So researchers here at Aberystwyth University, using equipment based in AberInnovation, were able to investigate extracting xylitol from wheat straw to see if this would be a valuable source, and to see if they could produce this in a more economical and eco-friendly way without using toxic chemicals, as well as investigating the scale-up of the process. So with this in mind, other sources of revenue have become attracted to farmers in recent years with wheat straw, and it has been used for energy production and also for packaging. But obviously more recently, Aberystwyth University, working through the Beacon programme, investigated its use to produce xylitol. So xylitol is a naturally occurring sweetener, and unlike sugar, it does not rot teeth, and it is not responsible for increases in diabetes or obesity. And for these reasons, it has considerable interest from food and biopharmaceutical sectors. So xylitose is made from xylose, which is found in plant cell walls, particularly woody plants, and birch is usually the main source. So, however, unfortunately, the use of birch trees involves harvesting the bark of the birch trees, which can effectively kill them. So more sustainable and more eco-friendly methods are needed. So researchers here at Aberystwyth, using equipment which is now in AberInnovation, were able to investigate extracting that xylitol from wheat straw to see if it could be a more valuable source. And I'm just going to show you a little bit about the system that they used. So raw materials, wheat straw, were brought into the facility. They were processed through our Biorefining Centre, and they're chopped up and put into the steam explosion rig here. to produce this pressed liquor, and then built up into a bulk liquor before going through this cross filtration system here. So this was to give an optimum concentration for the following fermentation process. And then the fermentation was done at several scales. So here at a bench top 1 litre fermentation, and then following up with 30, 50 and finally 150 litre fermenters. And it is worth mentioning here also that interestingly, and key to this process was the fact that the yeast from the gut of a click beetle was used specifically to convert the xylose extracted from the wheat straw into xylitol. So what was the outcome from this work? Well, a new, highly efficient and eco-friendly process for producing xylitol was developed. And from this work, a spin-out company, Architect Bio Limited, which is a member of AberInnovation and resides in our offices here, was formed in 2018. And they are now scaling up beyond that programme, Beacon programme's capabilities with Innovate UK funding. So also as a result of this work, Aberystwyth University and partners have been successful in winning a Newton Vava Fund Industrial Waste Challenge funding. This project aims to transform waste streams from the Indian sugarcane industry. That waste, which is known as bagasse, illustrated here, and transforming that into a range of valuable products, of which xylitol is one. And it's worth noting too that here the value that this could release in terms of potential xylitol market is illustrated here. It's been predicted that the xylitol market from bagasse could be worth to £1 billion by 2025. So moving on to the next case study, looking at food waste. So this is a problem that Corn approached the Beacon team with, and the corn manufacturing process generates a water-based effluent, so approximately 25 to 30,000 litres per hour, which is an enormous amount of liquid. And as their production of corn was predicted to increase, so too the quantity of that waste was predicted to increase, and they calculated to a potential of 1 billion litres per annum. And as their current water costs were around £2 million per annum, then this was going to rise significantly. And it was also recognised there are up to 1.5% solids in this wastewater, which also represented a significant loss of value. And this wastewater was known to contain compounds called nucleotide and glutamates, or NAGs for short, and some rare sugars. So these were evaluated and found to be a potential for use as a component of natural flavouring systems. So Corn really wanted to know, can we remove the water using an economically viable process? So the proposed project was designed to evaluate a cost-effective way of concentrating the effluent and using enzymes to maximise NAGs to deliver a highly potent flavouring system. So using our facilities here, the equipment within the Biorefining Centre, a waste stream filtration process was set up. And to do this, our daisy chain filtration rig here was used, and the liquor was passed from a microfiltration through on this end of the equipment through to reverse osmosis at the other end. And this meant passing the liquor first through microfiltration, which is typically used for removing things such as yeast, I suppose larger particles from the liquor, and then passing this through ultrafiltration, which removes slightly smaller particles, so in this case proteins, which are smaller, and then nanofiltration, so smaller and smaller particles removed with each filtration level. to eventually the reverse osmosis, which is illustrated in here. This allowed water then to be extracted. So the reverse osmosis retained liquor, so in here, in this section here, was then passed over a spray dryer, and the resulting solids from this were contained and were the desired nags. So these nags were then used in various recipes, including processed cheese, and then used in taste trials by Horn and also Waitrose. So as a result of that work, the waste put through this process produces the UK's first major source of NAGs, or nucleotide and glutamates, which are, as I said, well known for being taste enhancers. So this was a cost-effective method, and it produced the products that can be used as taste enhancers in foods such as cheese and ready meals, etc. And as I'm sure most people are aware, there is widespread concern about levels of salt consumption, and reducing salt, as here recommended by the World Health Organization, they have suggested that's of major importance. So the recipes tested reduced their salt content and replaced this with the taste enhancers. And the taste test showed that the salt content could be reduced in recipes by up to 30% with no effect on taste, so tremendous result. And also this work has led to a successful 2-year innovative project refining the process and its use as a taste enhancer. So my final case study is looking at added pharmaceutical value, and it's focusing on pharmaceuticals being produced from daffodils. So this is a collaborative project between Aberystwyth University, Harper Adams and Agroceutical Products, and use equipment obviously in the Biorefining Centre in AberInnovation. So what was the problem? Well, there are some issues with current production of galantamine, and galantamine is an approved drug for the treatment of Alzheimer's. It is synthesized chemically and is very expensive to produce, and its sales worldwide are in excess of around $500 million per annum. So sadly, its use is currently constrained by its production also. So researchers wanted to see if they could find a more cost-effective source of galantamine. And it's known that certain plants contain galantamine, and daffodils obviously were selected as the only possible economically feasible source in the UK. So in addition to increase the cost effectiveness, the researchers wanted to investigate whether they could be grown in less favoured upland farmland, and if extraction of other alkaloids could be considered. So how do we get from the field here, the daffodils, to the actual product here and at pilot scale? Well, there were 3 parts to this project really. This project tested a novel approach to producing galantamine based on sowing daffodils into existing upland pasture. This was carried out at Pwllheuyn, which is an Aberystwyth University upland research farm. Specialist machinery was developed at Harper Adams for planting the bulbs and for harvesting spring growth. So in spring, the green growth was harvested, and this material was then taken to the biorefining facilities to be processed. So just to explain that process, so I know here we have bulbs in this image and not the top growth, but initial trials were done using bulbs and compared with the green top growth and showed no difference in percentage content of galantamine. So harvesting the top growth means it can be harvested each year if grown in a farm setting. So the raw materials were chopped up. They went through enzyme digestion across here. Then the fibre separated off. The liquid was then put through centrifuge. and then to the daisy chain filtration, which I've mentioned in the previous case study, and where a concentrate of 23% solids was produced. And this liquid was then cleaned up and passed through high performance counter current chromatography instrument. And this identified other alkaloids through the system, and these were all separated out, and this crystallized product produced. So what they found was that this process is not really viable for the sole extraction of galanthamine from daffodils. So actually extracting other alkaloids, and these were alkaloids such as narcoclassine and hemanthamine, glycerine, made the process viable. And just to illustrate the importance of these other alkaloids, they all have potential cancer applications, and in some cases are already being used for preventing tumour growth. So the outcomes from this project, well, they showed that upland farm areas could offer a sustainable source of plant-derived galanthamine, but only if the additional alkaloids were also extracted. And following the harvest, it's interesting to note pasture was grazed with ewes and lambs, and it was found that incorporating daffodils into grazed pastures did not affect lamb performance. And also, this has shown the potential to increase the economic sustainability of farming communities, particularly in less favoured areas. And I guess this method provides a more sustainable source of galanthamine and other useful pharmaceuticals. So these are but a few of the really interesting projects we have been involved in. And now that we have our new facilities and our building is complete with equipment, including those mentioned in these case studies, we have a large list of other equipment there as well. There will be more opportunities to be involved in projects like these. So if anyone would like to know more and find out how to work with us, please do contact me on my contact details there. And I notice my lights just automatically switch off from lack of movement in the room, so it's obviously very good timing there. So I'd just like to say, diolch yn fawr, and thank you very much for listening.
Thank you, Rebecca. It's good to see you're sitting in a sustainable building, which turns the lights out. Fantastic. Thank you. No, and really good to see that level of investment and infrastructure going into West Wales as well. On that, there was a question from Mark about the sensory centre that you've got at the Institute. He's particularly interested with odours and sort of the air pollution aspect of measuring those. He wondered if well, first of all, the capability of the sensor facility, but also what's the focus of the sensors and the research that you're using there?
So food, fuel, and what was the other one? Food, fuel, and farming. Well, I suppose we look at farm to fork as one of the examples of work that we would like to have running in our AberInnovation Centre. Obviously, a lot of what we do focuses on areas of expertise in the university, and obviously there's a wide plethora of expertise there in different departments. So we have an Institute of Biological, Environmental and Rural Sciences, but we also have a computer science department, we have physics departments, and many other departments that also link in with projects that go on in the centre as well. So although we have that theme, there are other areas of expertise that can come into projects to work with industry in that sort of theme. Yeah.
And the sensors themselves, how do you develop sensors, or do you use sensors in that sort of infrastructure and equipment that you've got?
Sorry, when you talk about sensors?
Actual sensors, yes.
Oh,
I know that Mark's particularly interested in detecting odours with sensors.
Oh, okay, yes. So
I don't know if you can hear me. This is Mark here. I was just to say, I was just interested in what you said about a sensory centre, which I thought would refer to maybe smell or taste or something. I just wanted to know a little bit more about what your focus was. Thank you.
So the sensory panel will consist of people, obviously. for our sensory booth. So we will be training them in all aspects of taste analysis and so on. And I'm not completely au fait with all the equipment that's going in the building, but definitely anything to do with sense, it will be there.
Just a comment from Brian in the comments as well. He's going to contact you later about the biodigestible analysis facility and equipment. So he's obviously got an interest in that, which is good. So it's very fitting that the last case study you talked about was using daffodils, of course. I mean, was that a surprise?
I think it wasn't a surprise that galantamine is in daffodils, and I think that whole family that daffodils are in, you can find it. It's just, I guess, part of the problem was trying to find a way of extracting it or growing it in a way that was economical, and daffodils obviously being something that grows well in the UK and also in upland areas as well. That was why the focus on daffodils, I guess. So yeah, definitely increasingly lots of investigation into what plants contain and how the content those discoveries can assist or feed into all sorts of areas of human use. Definitely a lot of research going on with that, and we work with a lot of companies who also do the same in those areas.
Good. Well, thank you very much indeed, Rebecca. That's good. And we'll reconvene in the panel after our final talk this morning.